So, today we will still continuing the cytochrome C oxidase, in which we have seen last time that it has 4 metal ion centers. So, when a system, particularly a catalytic site which has 4 metal ion centers, it would be definitely a complicated system, and it is attached to the terminal enzyme in the respiratory chain So, we have, if it has the iron site, and the basic function of it we all know now, that if it can supply the electrons from the reduced form of cytochrome C. So, if it gives us 4 electron, which is very important, that we are talking about, that on the left hand side, if you have the reduced cytochrome C, which ultimately delivering electrons to our dioxygen molecule along with the consumption of 4 hydrogen ions. So, from the left, the cytochrome C oxidase is basically consuming 4 electrons, and thereby it oxidizing the cytochrome C site to its corresponding cytochrome COX, means cytochrome C oxidized form, plus 2 H2. So, that is the basic reaction what we are talking about involving cytochrome C. That means, how we can convert this O2, the mitochondrial O2 in the respiratory chain to 2 H2O, and identification of these centers are also pretty much difficult, and characterization of these ultimately tells us that we have the heme site, the iron porphyrin site, as well as we have the copper site, and these heme sites are 2 different. One is A, another is A3, and 2 copper sites are also different, and optical rotatory dispersion and circular dichroism techniques, not only EPR or other physical techniques, but also sometimes some other techniques are also involved there to identify the different environments. That means, all of them have the different environments. So, to find out the environments are important, and as we all know now, when we talk about something, that means the environment around any metal center, which may be catalytic, which may not be catalytic, but to identify the exact environment is very difficult sometime, because we have the coordination environment. That means, the ligand what we have in our hand, and when we identify this heme, we know that the ligand is your porphyrin. And when we differentiate these two between A and A3, your coordination environment must be different. How do we know that? If we get something and if we have some idea that this heme A and heme A3, that means one is cytochrome A, that is Fe, this is the porphyrin plane, and we have two nitrogens. That means it is coordinatively saturated, that means it is octahedral and 6 coordinate. So, it will show a typical corresponding electronic spectral pattern as well as your ORD or CD pattern, which are completely different from our A3, where A3 is nothing but the same iron attached to porphyrin with one nitrogen. That means it is now pentacoordinated. So, when we get a pentacoordinated one, spectral feature will also be different. And also, if we are able to identify that this particular vacant site, so this is the vacancy, and this vacancy can bind O2 molecule. So, O2 can be a very good ligand for this site, or it can bind carbon monoxide. So, this is the right information what we can have when we are talking about the environment in terms of its coordination structure or coordination environment. Then when it is binds to CO, it gives some typical characteristic feature, and we all will be interested to know, because there are several things involving over these two sites. That means, one is this iron site, which is nitrogen from, we all know now these are all obviously the histidine nitrogen, this is also your histidine nitrogen and the vacancy. So, this second one, what is that? That I will come to that point, but before that, if we find that in your hand the cytochrome C oxidase in its two different forms, how it is that? That means two different forms mean these two iron can have +2 as well as +3 oxidation state, and we all know the spectral features, even that corresponding irreversible spectra or the ligand field spectra, these two are different for iron 2 as well as iron 3, and not only in the +2 or +3 oxidation state, but they also differ in high spin state as well as the low spin state. So, if we are able to find out the corresponding basic lambda max values for the oxidized form and the reduced form, and those are sometimes very much characteristic, because in this particular case in the visible range it shows a band at 599 nanometer, then 2 or 3 bands in the range of 424 to 417 nanometer, that means that high energy range. So, these two are definitely like the porphyrin band, that means what we know that as the solid band in some cases, the 450 band for some of the cytochromes. So, these are high energy band and one low energy band at 599 nanometer. So, in the reduced form we get some other bands which are at 605 nanometer and 444 nanometer. So, this you can have, that means you can identify immediately looking at the spectral characteristics for the oxidized form of the cytochrome C oxidase as well as the reduced form of the cytochrome C oxidase. That means, in oxidized form we already know that you have the 2 heme center and 2 copper center. So, in the oxidized form, that means when we talk that they are fully oxidized. So, there are 4 electron transfer in the system. So, this 4 electron if it is there, that means one after another, that the system which is reduced by 4 electron, that means which is super reduced like iron sulfur proteins. So, fully reduced form have some characteristic feature and fully oxidized form have some characteristic feature. So, in between you can have 1 electron reduced product, 2 electron reduced product, 3 electron reduced product. So, you can, based on these spectral characteristics, you can identify the fully oxidized form, the fully reduced form, or 1 electron reduced, 2 electron reduced, or 3 electron reduced form. So, not only these characterizations based on UV visible spectra, but sometime we can have some idea about the same cytochrome C oxidized, but this is now through this, that means binding at this iron by carbon monoxide, because we all know that when it binds to carbon monoxide it has different spectral properties. So, in the reduced form, first we will check whether it will bind carbon monoxide or not. If it binds that carbon monoxide, that immediately tells us what? That immediately tells us that this is the site, that means your heme A3 is involved in carbon monoxide binding, and the spectral feature related to this particular iron site is getting modified, and this particular one we have the spectral positions like 605 or 444, they are lowered in intensity compared to this reduced cytochrome C oxidized. So, they are lowered in intensity, whereas the other signatures, that means the corresponding lambda max values at 590 and 431, they remain unchanged. So, if we can have a database like this, that means if we can have all the spectral feature for the oxidized form, reduced form, as well as the intermediate one. So, it would be very easy for us to identify these particular forms, because in these two cases, that means if we have the oxidized form which is 6 coordinate and the reduced form, we know it can have the low spin form, or in the reduced form it can also have the low spin form, because these are the most predominating form for hexacoordinated iron site for heme A. So, you have S is equal to half and reduced form S is equal to 0. Similarly, for A3 site which is oxidized as well as reduced, and interestingly, since it is 5 coordinated one like myoglobin and hemoglobin, this can be in equilibrium between these two spin state, that means the high spin as well as low spin, that means you can have spin value 5 by 2 or half integral, but the reduced one will have a spin value equal to 2. So, this is making a situation very much complicated. If we cannot see one after another, it will be very much difficult to identify the actual iron site as well as the other metal ion, which is also paramagnetic in nature, and it can also provide unpaired electron to the system. So, this particular one, that means if you have the iron sites and the copper site, and both of them can be in the oxidized form as well as in the reduced form, and these are sometime if we can find that they are interacting to each other. So, that identified afterwards, that means one of the site which is paramagnetic in nature and this is also paramagnetic in nature. So, positioning of these two sites is very important, and until and unless we have the corresponding X-ray structures in our hand, we cannot say that how far they are. That means, whether these two sites are 13 angstrom apart or 30 angstrom apart, because these biological electron transfers are all the time they are going through a outer sphere mechanism, and in a long range, say 30 to 35 angstrom, because you have the protein backbone, the amide backbone you can have. So, outer sphere mechanism is operative in a very long distance, which is unlike for our molecular systems, where we need for a typical exchange. That means, if you have in this particular site one unpaired electron and on the right hand side you have one unpaired electron, if they are within in a molecular system within 2 to 3 angstrom, then only they can interact together. When they share a common orbital, we call them as a magnetic orbital. So, we all know that then you can have the corresponding ferromagnetic or antiferromagnetic interaction. So, looking at these values particularly, that if you have iron, because we have just started our discussion for the iron sites, and there are two different oxidation states and the two different spin states. So, we can have some basic idea, that means what are the number of electrons present on this iron and the number of electron present on this particular site. That means, whether some of these cases they will be EPR active or EPR inactive, and in the background you will have the corresponding spectral characterizations. Okay. So, during this reduction process that we have seen earlier, that if you have one site and we designate that site as copper A site, and the other one is our copper B site, and close to that copper B site we have heme A3. That means, the distance between the copper center and the iron center can be determined. Similarly, this copper A site, which is close to our heme A site. So, you have 1, 2, 3, 4 sites in your hand. 2 of them are with iron and 2 of them are with copper, and this copper A site is dinuclear, making the system much more complex, is not a mononuclear system. So, this particular copper A site, which is a dinuclear one, and if you just recall what we have on the left hand side, we are having cytochrome C, then you have the cytochrome C oxidized, and on the right hand side you have the dioxygen molecule. So, this particular copper A site basically receiving the electron from cytochrome C, it is receiving the electron, and which is therefore responsible for the oxidation of the 4 molecules of, when it is receiving the electron from cytochrome C, and this cytochrome C will be converted to cytochrome C ferric based, means cytochrome C 3 plus. So, when it gives that electron, and that electron if it goes to heme A site. So, if you can get one after another, that means you need not only the single electron transfer, but you need 4 electron transfer to the cytochrome C oxidase site. That means, the particular system is either a 4 electron oxidized form or a 4 electron reduced form, and 4 cytochrome C molecules are responsible for transferring electrons to convert your cytochromes, because the entire assembly is now known as your cytochrome C oxidase, and that cytochrome C oxidase is reduced by 4 electron reduction from the cytochrome C. So, these two now they are close to each other, and after reducing this center, electrons are ultimately pushed into this particular system of heme A3 and copper B, because this is our catalytic center. Why? Because it can bind the dioxygen molecule. So, you are getting at one hand the 4 number of electrons, the 4 electrons you are getting from this particular side, and you are having something where you have the dioxygen molecule which is attached to this catalytic center, and ultimately one after another, if you think that if we push those 4 electrons to the orbitals of O2, then you will be able to reduce that O2 to water molecule. So, this reduced form we get after we have the oxidized form of cytochrome C oxidized. So, this is oxidized. That means it is fully oxidized, and this fully oxidized form sometimes designated as O4, because these are some states basically, because biologists sometimes do not bother about the iron site and iron oxidation state, but they can identify the corresponding species in terms of its EPR activity, in terms of its electronic spectral activity or any other physical activity. So, the oxidized form, if we label it as O4, and because it is 4 electron oxidized form, so the number of electron transferred to the catalytic site. So, it can accept 4 electrons. So, the total number of electrons it can get, that means when it remove those electrons, you get the corresponding catalytic site both heme A3 and copper B. That means, if you have Fe A3 which is in the trivalent state and copper B in the bivalent state. So, this is our only option we can have. That means, the ferric site of A3 and cupric site for copper B, which is your catalytic center. And as just now we have seen that we can reduce these two. That means, you can have a fully oxidized form or you can have a fully reduced form, and in between you can supply 1 electron, 2 electrons or 3 electrons to that. So, if we can go for a reduction of 1 electron of this particular state and 1 electron of this particular state, we get one another state which is labeled as U1, and the second electron, that means U1 will go to R2. So, do not be confused with this, these are difficult level for this is not much, but it is the corresponding reductions. When we have the fully oxidized form, it is O4, 4 is the number of electron transfer. When you reduce it by single electron, it is E1, E1 is the number of electron transferred to O4. It is the corresponding number of electron, that means 2 electron has been transferred. So, it is the reduced form which is R2. But what actually happens there? In our case, that means we will see that this particular iron site will be converted to 2 plus. So, your A3 is converted to Fe2 plus and your copper B is converted to 1 plus. So, that is very important, because if we see there that these two are close to each other, that means you have the A3 So, another form is there, that means you have A3 which is Fe3, and just now we have seen that it can be in equilibrium between high spin and low spin, and you have a nearby copper B which is copper 2. So, now this gives us the some idea about that this particular center is paramagnetic, which is also EPR active, and this is also EPR active. But like your iron sulfur clusters, what we have seen, that sometimes you can have two sides and individually both of them are paramagnetic, but if they are strongly coupled to each other, depending upon your coupling, and we consider that their corresponding J values, how they strongly are coupled, and we try to record there the corresponding EPR spectra. And if it is a paramagnetic one, that means we are just nullifying some of these paramagnetic species, and sometimes we get that it is EPR silent, we do not get any signal, corresponding EPR signal. That means this is strongly coupled and the coupling is strongly anti-ferromagnetic. So, this gives some clear picture for this reduction to this particular species. That means, already we have seen that this particular heme A3 is 5 coordinated. So, it is 5 coordinated. So, it has some position which can bind your dioxygen molecule, and also we have seen that this particular copper B site was only bound to 3 nitrogen. So, it is not even 4 coordinated one. So, both the iron site as well as your copper site is coordinatively unsaturated, because it can have a maximum 6 coordination sites, this can also 4 coordination sites. So, we are pushing what? We are pushing just talking about this ligand, talking about the binding of dioxygen molecule. So, basically you have now the dioxygen molecule in your hand, and we are just simply dumping this dioxygen in the catalytic center, and you have iron, you have copper which can bind one more extra ligand, which can be your O2 molecule, which can be your oxide molecule, which can be your peroxide molecule, and if they are bridged, so antiferromagnetic interaction we find, and if it is through some bridge, that means bridge either the entire O2 molecule or its some reduced form, that means the superoxide or peroxide, or you can have, you will find it that nicely, that whether you can have the OO bond cleavage. So, if you have the OO bond cleavage, then you generate the oxo. So, it can also be a iron oxo copper site, and if it is a iron oxo copper site and if they are strongly coupled to each other, we get a system which should be EPR silent. So, these particular sites, when it goes from one after another, so starting from our O4, E1 and R2, we will find that what other species, because we are not talking about all the 4 electrons. So, you can have several sites for this catalytic endeavor. So, these are the different steps. So, then on the right you can have one more here, and one more. So, here is nothing but our O4, what we already seen, this particular O4 state. And O4 state we all know now is your Fe 3 plus, then copper 2 plus, Fe is your FeA3 and copper is your copper B, and this is bound to a water molecule, copper site, and this iron site is bound to a hydroxide group, and this particular one has also nearby YO minus. What is that YO minus? YO minus is another thing which we are bringing into the picture, is the organic part which can give rise to some unpaired electron to you, that is your tyrosine residue. So, this tyrosine residue which is coming from TYR, tyrosine 288, because we all know that a phenol unit can be oxidized. So, phenoxyl radical. So, the radical mechanism. So, it is not fully dependent on metal center redox activity, but it is also dependent on some organic redox activity. That means, the phenoxide radical is also involved over there. So, this YO minus is present, and this tyrosine residue is very interesting because it is covalently cross linked to one of the copper B ligands, because already we have seen just now that this copper B is bound to 3 nitrogens, this copper B, 1 nitrogen, 2nd nitrogen and 3rd nitrogen from the histidine residues, and that histidine residue is then covalently linked to this tyrosine group. So, this tyrosine group is not going away, that means, this should be close to your copper site. If it is covalently attached to your ligand site or around copper, that means, if you have the copper binding from 3 nitrogens of around this copper, so your tyrosine group is also nearby, and that tyrosine group through oxidation as well as deprotonation, because it will accept proton as well as accept electron after it has been oxidized. So, this can play some important role to play for the electron acceptance as well as electron donation. So, this particular O4 group we already seen there, that means, already we leveled that it can be reduced by a single electron to a E1 state. So, that E1 state, what is that then? This E1 state will be something where we just through this particular step we give electron, and electrons are not moving alone, because these all mitochondrial system, they are all membrane bound, and we have the phospholipid groups, and those phospholipid groups have the attached cytochrome C oxidase groups, and whenever we have electron transfer, so when you have the electron, either it is donated or accepted to the site, you have to have the side by proton transfer as well. So, from left hand side, if your electron is pushed into the system, the proton will come from the opposite side. So, the proton transfer will also take place over there, and some extra proton is also pumped to the system along with this electron transfer. So, this proton either it goes directly to the system, that means, in this particular case when proton is pushed over there, you have the deprotonated form of the tyrosine which is getting protonated. So, your YO minus is going to YOH. So, that protonation, always keep in mind that this particular case is a membrane bound thing. You should also think of the corresponding pH values. So, if your pH is something which is not matching with the corresponding pK value of the tyrosine residues, it will remain as its deprotonated form, and when it is little bit acidic, it goes to its corresponding protonated form. So, this thing and these two sites you have also. So, you have Fe site as well as the copper site, and now for the single electron transfer there will be a competition for the corresponding electron acceptance for the corresponding E0 value for the site, that whether your iron site will be reduced or the copper site will be reduced. So, as you move from one site to another, that means you have a corresponding potential gradient. So, in the first case it is your copper site which will be reduced. So, cupric copper will be converted to cuprous copper, and same iron you will have which is still bound to your OH function. So, this you get as your E1 state. Then we move for here, that means in this particular case you get the corresponding electron transfer again. So, E minus and H plus. So, the second electron which gives us the corresponding state which is known as, we already seen there is R2. The first electron E1, one electron transfer, this is the second electron transfer, and this particular case if you go there, go for that, this not only electron transfer as well as the proton transfer, this particular water molecule sometime go away when you reduce it from cupric to cuprous. So, in this particular case what is happening there, that you are losing also this particular water molecule attached to your cupric copper site. So, all these things, so that means when you have the already it is tri-coordinated and one loosely bound water molecule you have, and you are reducing it, the copper site is reduced to copper 1 and it is losing your water molecule, because all these coppers we know when we move from a system that we copper 1 always have a coordination preference for a tetrahedral geometry. So, if you have already a square planar one in this particular case, so it will simply move from a square planar to a tetrahedral geometry. And not only the immediate coordination environment for copper as well as iron, but you have in the second level you have the protein environment, and that protein environment due to the change in the coordination site for the copper is also getting little bit modified. So, when we move to your R2 state, and this particular case you have still YOH, that means the tyrosine in the protonated form, and now the second electron goes to your iron site. So, your Fe3 iron is now reduced to Fe2 iron, which is based on the heme A3, and you have copper B. So, this particular A3 heme protein now is now ready to accept, because we all know now that for the myoglobin or the hemoglobin system, that when you have the iron site in the reduced form and if it is pentacoordinated, then only it can accept the dioxygen molecule. So, this is now ready to bind this dioxygen molecule, and within a very small span of time, so 10 microsecond, within 10 microsecond it binds the dioxygen molecule to our heme A3. So, 2 electron reduction, and you have the pentacoordinated iron site in the reduced form, and which is utilized for dioxygen binding. So, within 10 microsecond you get the binding of O2, and this particular one means we have not pushed any extra electron to the system, but we have the binding of dioxygen molecule. So, R2 is going from one state to another state, which is now known as A2 state. So, in this particular A2 state we have YOH, the nearby tyrosine residue, then Fe2 plus, which is of A3 and which binds to dioxygen molecule and we have copper B in plus 1 state. So, this particular dioxygen binding, because afterwards it can give you something that if you have only the isolated pocket for dioxygen, that means if your other end, so this particular dioxygen molecule, so is not symmetrically bound between the iron side and the copper side. That means unlike your hemocyanin, in case of hemocyanin we all know that you have two copper sides and in between you have the dioxygen molecule and the dioxygen molecule is bridged by two of these copper sides. But if you have a terminal dioxygen binding and that terminal dioxygen binding, because when there is a terminal dioxygen binding to any of these sides, we all know that there is a partial charge separation. That means, this will be this oxygen which is directly attached to iron side will be delta positive and the other end will be delta negative. And if this delta negative side which has some extra electron density, which can not only attract your copper side, but it also can attract some of the sides where from it can get hydrogen bonding interactions. So, if you have some imidazole side chain from the protein chain, it can be stabilized by hydrogen bonding interactions. So, if those hydrogen bonding interactions are there, so this particular entity, that means the O2 is only attached to your iron side, is getting stabilized, and if this O is strongly engaged in hydrogen bonding to some imidazole side chain or anything, then there is a possibility for your OO bond cleavage. That means, heterolytic OO cleavage is also another possibility when you have binding for one of the oxygen to the iron side, that we all know that the Fenton type of reactivity. You have hydrogen peroxide. So, any hydrogen peroxide or any oxygen based system if you can have and which is bound to your iron side, it can go for a cleavage reaction and that cleavage reaction is taking place on your iron side and this iron can go for another oxidation state which is 4 plus. So, when it is going for a plus 4 oxidation state, your OO bond cleavage has taken place. That means, this we all know now is a well known species which is known as the corresponding ferryle species. So, you have the iron in the plus 4 oxidation state and this particular state you have this O is in the oxo form and this copper which is also oxidized in the. So, lot of change is taking place at this particular point which is attached to HO minus and the other one, this is the other part, that means your Y group is there. Now, it is forming a radical. So, this is your radical form. So, tyrosine in radical form is there and this is also a very fast process and formed within 50 microsecond. So, it is very difficult to identify all these species and for this particular step we get a corresponding O bond cleavage, that means O bond is broken for this particular system. So, basic idea behind this is that you have already the corresponding species, that means 4 electron transfer for cytochrome side to the dioxygen side and this dioxygen when you go for the cleavage reaction and you should be able to produce the corresponding water molecule. So, when this is attached to the next step and here again you get, because already we have consumed 2 electrons here and here. So, now you have another electron again and the proton to the system and in this particular case the proton and the electron which is we are pushing to the system, the electron goes to your tyrosine residue. That means, your oxidized form is moving back to your phenoxide ionic form. So, it is going back to YO minus form. Iron is present as Fe4 plus attached to a oxo form, and which is your A3 still, and your copper is in 2 plus, and this proton basically when we push this proton, it is forming water. So, this particular case, so we just simply now complete our cycle. How we can complete our cycle now? Because now we have to push again some electron as well as proton, and some extra proton is also pumped from there, and these electrons are coming from where? These electrons are coming from heme A and copper A. So, these electrons basically what we are giving to the system are accepted from heme A. So, heme A, the function of the heme A is nothing but it can accept the electron from the left hand side from the cytochrome C, and it can donate that electron at this step. Similarly, this particular one, that means the formation of this corresponding radical function is important, because this particular case the enzyme which extracts 3 electrons. So, enzyme which basically extracts 3 electron, how we are getting that? So, we are getting 3 electrons from metal ions. So, do not confuse that you are getting all 4 electrons from 4 metal centers, because you are having all 4 metal centers. So, immediate answer can be your that both the iron centers are involved for electron transfer and both the copper centers are involved for electron transfer, but it is not that. So, 2 from A3, that means when it is moving from ferrous to, that means +2 to +4 oxidation state, and 1 from copper B, that means when it is settling between +1 and +2, and the third one is from the redox active side chain, that means the tyrosine residue, because this sort of activity based on the redox active side chain is also known for some other important molecules. So, if we go back to the cycle again, we will see that when we have all these, that means if we consider that no, this is not forming. So, another alternative proposal was there, like the formation of the corresponding oxidized ligand form, because this is a nearby ligand, though this tyrosine oxygen is not directly attached to the metal center, but it has some potential for donation to the iron side or the copper side, but it is not attached, because it is in such a way it is fixed within the protein chain that it cannot come to the coordination distance to the iron side or to the copper side. So, they are the corresponding distant tyrosine residues. So, if we cannot go for the corresponding oxidized form of this tyrosine group, so it can only go for some deprotonation reaction, and in this particular case what should be our alternative proposal for that is, if you have Fe in 4 plus oxidation state which is attached to our oxo form, that means what we are getting from here now, without going for this radical intermediate. So, if we do not go for the radical intermediate, because some of these species where we are unable to identify the corresponding radical formation in this particular case, because the radical, we all know the radical can be very easily identified by EPR spectral characterization. It has a very standard G value at 2.0037. So, the presence of radical can also be very easily identified if we can simply go for the measurement in the solution state for this particular state, and if you can identify this particular one, because this is closely matching to our standard for the EPR measurement, diphenyl picaryl hydrazide. DPPH is the standard what we measure for electron paramagnetic resonance measurement. So, if we can go for this, that means the radical is not forming and your ferryle oxygen is present as the Fe4 plus O2 minus. So, what is your alternative form? Your alternative form is that you have the cuprous site. So, cuprous site should be oxidized over here immediately. So, you have Cu2 plus, which is attached to your HO minus, and this then after accepting, because this extra step for electron transfer we are not requiring, this is not required. When we avoid this particular path, we just simply go for this path, we simply go for the proton addition to that, and this proton addition will convert this copper to copper water species, and this particular one is known as a form which is P3. So, why we are talking so much about all these things is very important, because we will see in our next class that how we can generate this particular species, how easily we can generate, even in the case of your Fenton reagent. From your college days you are knowing all this, that when you have a ferric species, and in the ferric species in some little bit reducing environment you put some hydrogen peroxide. So, this hydrogen peroxide, if they are giving you the formation of some ferryle species, and not only gives us this particular informations that this ferryle species is forming and the facile electron transfer, because we are starting from a case where you have this particular case, this was basically 3 plus. So, between 2 plus, 3 plus and 4 plus. So, iron can move from a plus 2 oxidation state to a plus 3 oxidation state to a plus 4 oxidation state, and if we have some amount of confusion regarding another extra electron, that whether you are oxidizing a particular system by another extra electron, that means your tyrosine residue is getting oxidized or your iron site is getting oxidized. So, it is very difficult to identify, it is very difficult to establish that thing between another configuration where your iron site is in plus 5 oxidation state and your this particular radical form is not present. So, this will find in case of catalases and peroxidases, will find that since you have some nearby phenoxy radical, and if you have some very useful E0 value for this oxidation of this phenoxyl radical, because all we know that sometime, because the E0 value we can modify when your phenol unit in either in the protonated form or the unprotonated form, which is bound to the metal center. When it is bound to the metal center, you can modify the corresponding E0 value or the electrode potential. That means, you can oxidize that particular site at a lower oxidation potential. And if you can have some another E0 value for the oxidation of the nearby porphyrin ring. So, that will find that whether in some other combination that this tyrosine residue is not getting oxidized, but in case of catalases and peroxidases we will find that your porphyrin ring is getting oxidized. So, you have large number of unpaired electron over there. So, you have the CC double bond, and that CC double bonded backbone can be oxidized. So, your P in that particular case, P will be P plus. So, that is very important, and interestingly also when we go for this tetrahedral structure for this copper one and this tetrahedral structure, because we are pushing the electron from here and you are changing your copper side from a cupric side to a cuprous side. So, if we are able to change the environment around copper and this copper is close to that of our iron side, and what we are willing to get, that means we are willing to get such a situation or such a conformational arrangement around the protein environment for this entity, that this O2 should go and bind to your Fe2. So, during that transformation, this particular environment around your copper 2, that means the corresponding tertiary as well as quaternary structure, quaternary protein structures around this copper is getting modified, and due to that transformation what we will be getting is that your A3, that means the environment around your A3, this environment you have the corresponding exposure. So, you have certain positions remain vacant. So, this exposure is important. So, it exposes to our O2. So, this exposure is important. So, you see the simple coordination chemistry, that means the movement from square planar to a tetrahedral geometry around copper, and this copper is already bound to the protein structure. So, due to this transformation you are modifying the corresponding environment from a tertiary structure as well as the quaternary structure, and you have some avenue for exposure to dioxygen, and that dioxygen molecule is nicely bound to your iron center for its corresponding ferrile formation. So, immediate information for all these things is that how they are strongly coupled to each other, that means these two sides are close to each other, and how you can generate the ferrile for the different reactivities. So, not only for the next we will see that your peroxidase activity or the catalase activity, sometimes this oxygen will be a very good reagent for transferring this oxygen to some organic substrate for hydroxylation reaction. So, the generation of this ferrile iron is an important tool to go for your hydroxylation reaction for any organic substrate, even in our drug molecules which are hydrophobic in nature, and if you have a corresponding phenol end, that phenol end will be hydroxylated by using this particular thing, that means this particular reagent if you have iron and hydrogen peroxide and sometimes iron and your simple dioxygen molecule. Okay, thank you.